All right. So let's get started. So the PowerPoint from Monday that we need to finish off, we're going to start with that. And the end of that PowerPoint will be the end of the material for test one. And if you've been on our Blackboard site, you should have seen that already, in that I have another folder called test two now, which the PowerPoint that I made for today, the 23rd, is found in that folder. So anything that we go over starting at the 23rd PowerPoint won't be on test one. The rest of the stuff we cover here at the beginning will be at test one. And just as a little reminder of what we were talking about on Monday, we ended on chromatography, and we were starting to talk about ion exchange chromatography. And just before that, we were talking about purifying proteins and primary structure. Right. So just a little refresher, we ended on this slide, and we were talking about the different types of ion exchange. For ion exchange chromatography, you have two types: cation exchange and anion exchange. And the name is telling you what type of protein you are binding to, right? So cation exchange, cations are positive. They bind to positive proteins, so they have negative beads. Anion exchange binds to negative proteins, so they have positive beads. And again, the idea here is that in your column, you have these charged beads, and you put your mixture through your column. And depending on the type of charge on your protein, it will either stick to the column or go right through. So if this was like cation exchange, a positive protein will stick to the column, not move once it's on the column, while a negatively charged protein would go right through the column. Then, to remove the proteins that are stuck to your column, you have to add salt. A high salt concentration will go and outcompete your protein. So the salt will stick to the column better than the protein does, and then the protein would come off. And here are pictures of the common types of beads you use. So you have CM, and you have DEAE. A CM column is negatively charged, while DEAE is positively charged, right? So the CM column is used in cation exchange, DEAE is used in anion exchange. And let's start with this question here, and I do have a poll so I can see how people answer this, but I want you to try and tell me, if we have these 6 proteins and I run them through cation exchange, rank the order of elution. An elution means coming off column. So I want you to rank what order will these 6 proteins come off the column. And let me get that poll up. And the way that this poll works is that I have like 6 different questions, and it's just which one would elute first, second, third, fourth, fifth, and sixth. So try to get that ordering, and then once you have the order, answer the poll with your order, and we'll go from there. If you have questions, let me know. If the poll is in your way, feel free to minimize it and then reopen it once you have your answer. But yeah, let's start with this cation exchange. Yes, question?
Yeah, sorry. What does the PL stand for again?
Yeah. So PI is it's isoelectric point, and it's telling you at what pH you are neutral. Right. So take that information and what we talked about on Monday when we were talking about what is the charge of these different peptides, and see if you can relate that idea to the overall charge of a protein. So see if you can figure out if you have a low PI, does that mean at a pH of 7, you're more negative or more positive? Because these columns are usually run like at a pH of 7. So you're asking yourself, okay, does low PI mean I'm overall negative or positive? Does high PI mean I'm overall negative or positive? So that's the first thing you have to figure out. And if you're not sure, go back to your amino acid chart that has the PIs and look at, okay, what is the PI of a negative amino acid? What is the PI of a positive amino acid? And that should help you determine the overall charge of these proteins. And we'll do about 1 more minute of this. So if you're not sure, take your best educated guess at this point. right. Oops. So let's take a look at this, and it seems for the most part we're in two minds. So let's go over what PI means again. All right. So PI is at what pH you are overall neutral, or what pH you have no charge at. So at low pHs, at a low pH, there is a lot of hydrogen in solution. And at low pHs, negative amino acids become neutral. So if you have a very low PI, what that means is that you have a lot of negative amino acids. It takes a very low pH for you to become neutral. So low PI means negative amino acids. While the opposite is if you have a very high PI, you have a lot of positive amino acids. And then if you have a PI around 7 or 8, you're already neutral. So you have like an even mix. So we're looking at cation exchange. Cation exchange means I bind positive amino acids and I do not bind negative amino acids. So the order of elution will go your most negative amino acid to your most positive amino acids, or proteins rather, not amino acids. So your most negative protein is the one with the lowest PI. So it should go 4, then 6, then 1, then 3, 5, and 2. So that's for cation exchange. Anion exchange is the exact opposite idea, where an anion exchange, I bind negative amino acids or proteins really strongly, and I do not bind positive proteins. So the order of elution is completely opposite. So in an anion exchange column, since we have positive B's, the first to elute would be protein 2, then protein 5, then 3, then 1, then 6, then 4. So that's your relationship between PI or pKa as well. There. So in proteins, we're talking about like what's the pKa of a side chain group, right? And here we're talking about PI. So they're slightly different topics, but they're related because they're doing with charge and gaining or losing a proton. But yeah, PI, what pH that you're neutral, and then how does that relate to columns? What's going to come off first, and how we can predict that based on PI. So any questions about that idea presented here, either about PI or either about column chromatography itself, anything at all? Will questions on the test be similar to this? Yeah, so you might see a question similar to this. I'm not saying it's going to be this, but yeah, a question like this would be 100% fair on the test. Like, I don't think this question's like, I think this question's good in that it's testing multiple things. And I don't think it's, if you understand it, it's one of those questions that you can answer relatively quickly. So I think, and if you don't know it, it's very hard to guess the right answer. So I think this is a great test question. Like questions like these are the type of test questions I like. All right. Let's go on to our next type of chromatography. And this is called hydrophobic chromatography. The basic idea here is the same thing, where you have a bead and these beads will bind to proteins based on their chemical properties. The chemical property we're looking at here is how hydrophobic your protein is. So on this image on the bottom left here, the red is your bead, and we have 4 different proteins. And these proteins are just showing you how hydrophobic each protein is. So here at the top, you have the most hydrophobic protein because they have 4 hydrophobic parts. Here, this protein at the bottom is the least hydrophobic protein because it has 3 hydrophilic parts. And the idea here is that you put your proteins on this column. And this is actually the opposite process where low salt concentration is used to kick off proteins, right? So when you add your proteins to the solid matrix, to the column, the hydrophobic parts of your protein will bind your beads. The higher the salt concentration, the stronger the binding. And that's one of those really weird chemical properties that we're not going to talk about in this class, but basically high salt concentration makes hydrophobic groups bind stronger. Then you remove the salt and proteins will start coming off, where the least hydrophobic protein will come off first and your most hydrophobic protein will come off last. The beads here are relatively simple. They're just hydrocarbons. And here is another question that tests one, do you know your amino acids and which properties they have? 2, do you understand this idea of hydrophobic column? So I do have another poll for this. It's the same idea. I want you to tell me, based on our protein sequences up there, rank the order of evolution in a hydrophobic column. So see if you can do that. See, this is a good way to refresh yourself on the 1-letter codes of amino acids here. So you're asking yourself which protein is full of hydrophilic groups, Which protein's full of hydrophobic groups? And I'm just going to list our different amino acids based on their properties right now. So hydrophobic. All right. So I listed all of our amino acids based on if they're hydrophobic, polar, positive charge, or negative charge. For some, tyrosine and histine, I put a little tilde there because there's arguments for tyrosine, if it's polar or not, because it does have a giant hydrocarbon ring and then one little OH group. So it kind of is both polar and hydrophobic. Histidine, both polar and positive charge. Histine's really based on a ratio, like 40% of the time it's positive charge, 60% of the time it's not. So there are some arguments for, like, which amino acids are truly polar. But in general, that's a good listing right here. So the way to really do this is just to list over and say, which amino acids in our proteins are positively charged or polar. So I'm just going to underline those. So we have R, K, D, S, T, and then H and E. So protein 1 has a lot of charge in polar groups. So 1, 2, 3, 4, 5, 6 has roughly 7 of them. And then protein 2, cysteine kind of, serine kind of, R, D, S, S, K, K, N. So this is 1, 2, 3, 4, 5, 6, 7, 8, 9 of them, roughly. And then protein 3, histine, R, Y kind of, and that's it. 3. All right. So based on that, the ordering itself, if you just look at polar and charge groups, the most polar / charged amino acid should come out first. So based on that, it would go 2, 1, 3. Yeah, I mean, the reason why I hesitated right there is there is some argument that cysteine is weakly polar, serine weakly polar as well. So when we actually do if we did this experiment, I could see it possibly coming out as 1, 2, 3, just because number one has a lot of charges like R, K, D, E, but probably just by looking at the polar groups, it would go 2, 1, 3. And to answer the question that many of you might be thinking, how would I answer this on a test? On if I give you a question like this on a test, I will probably pick the really polar amino acids. So what I mean by that is like N for sure is really polar, Q for sure is really polar, T is really polar, S, C, and Y are slightly polar. So like on a test situation, instead of S's here, I would probably just make them N's or Q's. And then you would say, oh, 2 for sure. But yeah, in general, to answer a question like this, just count up the polar/charge groups. Whatever protein has the most will come off first in a hydrophobic column. So questions about hydrophobic column or how to just look at the sequence and determine a protein's overall hydrophobicity. Right? Now, we're going to continue on with our journey into columns. And the next one is a gel filtration column. What a gel filtration column does is that it separates proteins by sizes. And it does that because the matrix of a gel filtration column is full of beads. And inside these beads are little channels that are shown here. And the idea with this is that small molecules will go through the beads. Big molecules don't. So the big molecules just ignore the beads and go straight through, while these small molecules, they go through the inside of a bead and they travel through the inside of the bead. Therefore, it takes them longer to go to the through the column. An example of this could be like, let's say you have like a massive hallway and you are trying to go to the end of this hallway. And you have two particles, particle 1 and particle 2. Particle 1 cannot go into these side rooms, so it just goes all the way through. It has no choice. Particle 2 can go into the side room, so there is chance that it will just go through the side room, and it'll take longer to go through the hallway and hence come out second. That's the same idea with gel filtration. So rather simple ordering here, but I do have the poll to go with that. Tell me, based on gel filtration and the 4 proteins I put there, what is the order of elution? Do about 30 more seconds here. Would you remind us what KD is again? Is it mass related? KD stands for kilodalton, and it's a measurement of weight for atoms. So 1 Dalton equals the weight of 1 proton. In kilodaltons, 1 kilodalton is 1,000 daltons. And so when you talk about weights of proteins, you talk about them in terms of kilodaltons. All right. And looks like you all crushed this one. So the heaviest protein is protein 2, 135 kilodaltons. So that would come out first, then protein 3, then protein 4, and then protein 1. So that's the order of elution. Some things to keep in mind with gel filtration. There's usually a range of separation based on the beads. So for example, when you're like buying beads, they have different fractionation ranges where it would say like, this bead is good for separating proteins that are 5 kilodaltons and maybe like 50 kilodaltons. Anything bigger than 50, they come out at the same time. Anything smaller than 5, they come out at the same time as well. So there's usually a range that beads are able to separate. So going back to our topic we talked about on Monday, let's say that you busted open an E. coli, right? And you want to know, and you want to try to separate it from the other proteins. Well, if you know your protein that you want to separate is like maybe 35 kilodaltons, then you would use a bead like this, because anything bigger than 50, you don't want anyways, so it's okay that they come out at the same time. Anything smaller than 5, again, your target's 35, so you don't care about keeping anything smaller than 5, so you're fine with them coming out at the same time. While 5 to 50 would be a nice fractionation range where, but what I mean by that is that they'll come out in like an interval where you could plot like time. Actually, it's not time, it's volume versus size, where it's volume of liquid that you add and size of protein, and it will be a nice like linear line where, if your target is 35, oh, after this amount of solvent is added to my column, proteins that are 35 kilodalton will come out. So I know when I'm doing this experiment, I want to keep the tube that corresponds to when 35 kilodalton protein should come out, right? So that's the really useful thing about this technique and about the beads in general, how to pick them. You have to make sure that your protein, if you already know the weight, pick a bead that can separate that weight. So that is gel filtration, another type of chromatography. That brings us, I believe, to our last type of chromatography, which is the best type of chromatography that you can do. It's called affinity chromatography. Usually, not always, but usually, if you can, you want to use affinity chromatography to separate your protein. And the reason for that is because the solid portion of an affinity chromatography are made to bind your protein and your protein only. And it does this through non-covalent interactions. And there's 2 general types of affinity chromatography. You have immunoaffinity, and then you have affinity chromatography using metals. So let's talk about immunoaffinity. Here, your solid portion of the column is made out of antibodies. And antibodies are extremely specific in that antibodies bind one thing and they bind it very well. So if you make an antibody against your protein, so you have to like special order this, by the way. And what you do is that like you ask a company to like, I have a protein, I want to make an antibody against it. And they'll do that by injecting it into like a rabbit or a mouse. And it costs a couple $1,000s. It's not cheap, but they'll create an antibody that's specific for your protein only. You then put these antibodies on beads, and after you bust open an E. coli, you pour all the proteins through this column, and only your protein will stick to the beads. Everything else will come off. The only problem is your antibodies stick to your protein very strongly. And so to get your protein off, you actually have to cut the antibodies off of the bead. So affinity columns are usually one-use items, and they're expensive to run, but they are very, very, very good at getting your protein pure, to get all the other proteins washed off. A cheaper way to do affinity chromatography, and one that is used in labs all over the world all the time, is using metals. Now, usually here you have either zinc or nickel being your solid state. And what zinc/nickel will do is that they bind to histidines, but they will only bind like if there's multiple histidines in a row. Like I'm talking about like 6 or so. This is called a hist tag. And in nature, having 6 histidines in a row like does not happen by chance. There's like a 0% chance if you look at all the proteins in the world that you'll just have histine, 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 histine. Just doesn't occur in nature. So what you do if you want to use a metal column is that you engineer your protein to have what's called a hist tag, to have these multiple histidines at either the end or C terminus generally. That's where you want to put them. And I'll just answer that question. You do that through our genetic engineering we talked about, where you just add 6 histidines, the DNA sequence of 6 histidines, either at the beginning or end of your protein, and you put that into your plasmid. You then, after that's created, you then run your sample through the column, and your protein will stick to the metal because it has those histidines, and everything else will be washed out. Then you just use salt to outcompete your protein. So you add high concentrations of salt, and your histidines will unbind to the metal and come through the column. After that, you usually want to chop off the histidines. So after you have purified your protein and you have kicked your protein off the column, you generally want to remove that hist tag because that hist tag is non-natural. And so you don't want to do experiments with a hist tag on there if you can help it. But that is affinity chromatography. You're using ligands to bind your molecule incredibly tightly, and it'll only bind your molecule. So any questions about the idea of affinity chromatography? All right. If not, we can move on then. I believe that's our last column. Now, one thing that I haven't mentioned yet, is how do you know when your protein is coming off the column, or any protein whatsoever? Because the vast majority of proteins don't have a color. They're clear in a liquid form. So it looks like water. So how are you supposed to know if a protein's in there? And to determine when a protein is coming off a column, or proteins, or how to determine protein concentration in general, we use absorbance. And we use the idea that tryptophan absorbs at 280 nanomolars. So tryptophan will absorb light at 280 very strongly. Tyrosine also does this, but very weakly. Phenylalanine absorbs very weakly at 260. So we really care about tryptophan at 280. And then we can use Beer's law, absorbance equals extinction coefficient. This is calculated for each protein. And to get extinction coefficient, you just read your primary sequence into a computer program, and this program will estimate the extinction coefficient for you. So that's a known. Concentration multiplied by path length. So path length is like always 1. And based on your absorbance at 280, you can determine the concentration of your protein sample. However, this only works if your proteins have tryptophan in them. If your proteins don't have any tryptophan, you have to find some other way to measure concentration, because this only works for tryptophan. Another complication with this method is that DNA absorbs at 260 and can also absorb a little bit at 280. So you have to account for any possible DNA contamination. So you usually also measure the absorbance at 260 to see if you have strong contamination of DNA. This last bullet point here, if it has a chromophore, you can measure absorption in that region as well. Your example there would be like GFP, right? We talked about how GFP can glow if you shoot certain light at it. It can do that because it has what's called a chromophore. A chromophore is just a part of a protein that can absorb and emit light. So if you know your protein has a chromophore already, you don't need to measure at 280. You can just measure at whatever the absorbance of that chromophore is. So usually when you do column chromatography for real, at the end of your column where you're collecting drips in a test tube, you'll have a laser that's shooting through these drips, and then you'll measure absorbance right across from that laser. So you shoot a laser at 280, and then this machine will register, okay, how much of that light was absorbed by each of these strips, and you relate that to which samples have protein or not. And this is all automated at this point. What you do in reality is you just load your sample into a machine, and then you let it run overnight. And you come in the morning and you're done. You just read the absorbance and you know which test tubes to take, which test tubes have my protein in it. But any questions about that idea or anything we've talked about so far? Right. So let's say you did some type of chromatography and you have a test tube full of protein. Usually that is not good enough to say that that test tube has your protein. So to determine that after column chromatography, the test tube you have actually has the protein that you want, there's different techniques you can do to 100% verify that you have purified your protein. One of these is called an ELISA. Another one's called a Western blot, but we're not going to look at that right now. We're going to look at an ELISA, which stands for enzyme linked immunosorbent assay. So the idea of an ELISA is that you have some like membrane. You have some piece of paper. And on this piece of paper, you have antibodies. And these antibodies will bind to your protein and only your protein. So the idea is you take your test tube that contains protein after column chromatography, and you put it on this piece of paper. If your sample contains your protein, it will bind to the antibody. If it doesn't contain your protein, then nothing will bind. So you put your sample on there, and here we do have protein that we want, that is our target, so it binds to our antibody. After that, we add a secondary antibody. This secondary antibody will only bind to your protein that is bound to your enzyme or your primary antibody. And this secondary antibody also has an enzyme. This enzyme will do some kind of chemical reaction that we can see with our eyes. So we add this secondary antibody and it will bind if our protein of interest is already bound. We then wash our membrane, so any things that are lightly bound or any contaminants is washed away. And then we add some substrate. And this substrate will interact with the enzyme on the secondary antibody and make some kind of product. And this product has to be visible to us. So it can either be like an actual color we see, like the visible product will just be some color we can see right on the filter, or it could be something we see after we shine a light on it that will give fluorescence. But the idea is like, if you put your sample here, you might get like a dark bands on your ELISA. That is showing you, oh, okay, so we have 5 dark bands here. That means our secondary antibody has bound to these 5 spots and are making our detectable product. And this secondary antibody only bound because our enzyme, or sorry, our target protein is found there. So we would know, okay, our target protein is visible or has been collected because we have a visible response from our ELISA. So that, in essence, is how an ELISA works. ELISAs were also used during for COVID testing. That's one of the ways you can really tell if you have like COVID. You just run an ELISA that is specific for a COVID-19 protein, and if your ELISA comes back and you get a signal on your ELISA, that means 100% sure you have COVID-19 protein inside you. So if during the news last year you heard the word ELISA ever mentioned, this is what they're talking about. So any questions about the ELISA technique? Anything you want me to go over again? Part that you might have been lost you at, or anything at all? Whoops, did not mean to write that. All right. And we have two more techniques that are going to be covered on test one. The first one is SDSPAGE, sodium dodecyl sulfate polyacrylamide gel electrophoresis. Don't worry, I'm not going to test you on if you know what each letter means. What SDSPAGE is a way to separate proteins by mass. And this is another way to determine, after column chromatography, if the sample you have is pure. And we can take a look at this on the image on the left here. So let's say we did an experiment. And I'm just going to focus on the columns I circled here. So column 10 is called a ladder. And it's just a sample of known molecular weights that you add to your gel as a way to know what each band's weight is. Right? So 10 is something you add to the experiment so you can measure weights. Let's say that we are trying to purify a protein, and we open up the E. coli and we do salting out. So let's say our first thing is salting out. That's the first experiment we do. And we want to know, okay, after salting out, how pure is our protein? And let's say we run our salting out on columns 11 and 12, right? So on our page, after our salting out experiment, we put the results on columns 11 and 12. And you can see there are a lot of different proteins. Each one of these lines is a separate protein. So after salting out, we still have contamination. We have a lot of proteins in here, and our goal is to only get one protein through our purification scheme. So let's say, okay, after salting out, I'm going to do affinity chromatography now. And affinity chromatography, let's say that's in rows 13 and 14. You can see after 13 and 14, or on 13 and 14, I only have one protein that's showing up. So what that is telling us is that our sample is pure. We only have one type of protein in there. And then what you could do is you could do ELISA. So the sample from affinity chromatography, you just showed on a page gel, there's only one protein in it. You could then send that same sample to ELISA and say, okay, is this protein that's in this gel my protein? And ELISA could confirm that for you. So SDSPAGE is really just a way to see if your samples are pure or not, or determine, is this actually my protein? The way it separates proteins by mass is that you add a detergent called SDS. That's where it gets its name from. And you also add 2-mercaptoethanol, or also called beta-ME. So what SDS does is that it binds to all proteins non-specifically. So for it's like for every 1.7 amino acids, you will have one SDS molecule bound. And it doesn't matter what your amino acids are. For every protein, SDS binds the same way. And SDS does two things. One, it denatures your protein. Two, it gives it a negative charge. So overall, your protein becomes denatured, so it's no longer folded, and it has this overall very negative charge. And then you add beta-mercaptoethanol, which breaks disulfide bonds. Who wants to remind us what amino acid forms disulfide bonds again? I believe we talked about that on Monday. What amino acid breaks disulfide bonds? Or sorry, what amino acid forms disulfide bonds rather? Cysteine, very good, yes. Cysteine forms disulfide bonds. And when we do SDSPAGE, we don't want to have disulfide bonds because we only care about primary structure. And disulfide bonds will make your protein look bigger than it actually is. So we don't want that. So we break all those. Then the SDS, or the PAGE, so the acrylamide gel. What acrylamide gel is, when you look at it like underneath a microscope, it basically looks like this. It's just a web of acrylamide, and acrylamide's a polymer. So it's a web of chemicals. And big proteins get stuck in this web and they don't move very far down. Small proteins can go through the holes in this web, so they move down the farthest. So small proteins go down far, big proteins are at the top. Don't get this confused with gel filtration. Gel filtration is the opposite. Big proteins come out first, small proteins come out last. SDSPAGE, small come out first, big come out last. And the way we move these proteins through this gel is that you have an electrical charge. So at the bottom here of this gel, you have positive charge. At the top, you have negative charge through electricity. And so since your proteins are all negatively charged because of the SDS, they will naturally move towards the positive end when you have the electricity on. Small ones will go down farther than big ones. So that's SDS in a nutshell. Just to make sure we have it, I have this quick poll again. Tell me, based on the sizes of proteins I have on this question, list them in order from which one's going to travel the farthest down the gel all the way to which protein is going to be at the top of the gel based on what I just said about SDSPAGE in the last few minutes. Do about 30 more seconds. So if you haven't answered yet, please do so. All right, so it looks like we pretty much have that down. The one that's going to move the farthest is the smallest one, then the second farthest, third least. So the complete opposite of our gel filtration there. Any questions about the theory behind SDSPAGE or anything at all? All right. The last technique we're going to talk about, that's going to be on test one, is called 2D gel electrophoresis. So let's talk about what 2D gel electrophoresis is. In 2D gel electrophoresis, we are separating proteins rather by two different aspects. One, we're separating by size. Two, we're separating by their PI. And the idea here is that on this gel, going from left to right, we have like, imagine that you would split this gel into like 14 different sections, right? So on and so forth. And each section is a different pH. So in this section, the pH of the gel is 1. This section, the pH of the gel is 2, all the way down to the pH of this gel is 14. So question, SDS just gives every protein a negative charge, so it'll go down the gel. It does that, and it unfolds the protein. So normally proteins have like some kind of shape to them. What SDS does is that it turns every one of them into like denatured spaghetti, because when going through the gel matrix, like I drew, it's just random holes everywhere. And so if your protein still has shape to it, then it could get stuck at random places due to its shape. However, when you unfold everything looks the same now. They all look like spaghetti, more or less. And so they're traveling through the holes is strictly based on their size now, not their shape. So SDS makes everything negative, and it makes everything unfolded. So it does two things. All right, so getting back to this idea of 2D gel electrophoresis, like I was saying, each section of the gel has a different pH. And the first thing you do is that you do this isoelectric focusing, where you give an electrical charge going from left to right. And who wants to guess that what happens when a protein, well, actually, let me restate this sentence. Who wants to remind us what happens when a protein enters an environment where the pH is equal to its PI? So what happens if I have a protein and I put it in a solution where the pH of that solution is the same PI as the protein? What happens to the protein when charge-wise? What is the charge of that protein? It becomes neutral, correct. All right, so if I'm in an electrical field, and I'm neutral, I don't have any charge, do I move in that field or not? Like if I have positive charge on one side of me and negative charge on the other side of me, but I don't have any charge, do I move? You don't. Right, correct. So the idea here is that first, moving from left to right in electrical charge, a protein will stop moving in this gel when pH equals Pi. Because you're no longer charged, and so you're no longer affected by an electrical field. That's called isoelectric focusing. Then, after that's done, you stop the isoelectrical focusing, and then you do SDSPAGE, where you move from top to bottom. And now you are separating by size. So by doing this technique, let's imagine that you discovered, or you're studying a cell, all right? And you want to know what proteins are in this cell under different conditions. You would do 2D electrophoresis where you would bust open the cell, take all the proteins, run them on this type of gel, and then you could say, okay, I have a protein right here. I'm going to take it off the gel and I'm going to discover what it is. Okay, I have this protein that's also in the cell. I'm going to discover what it is. It's a way to separate lots of proteins at the same time to try and discover what they are. So it's a very fast way to separate a lot of proteins. So you separate by charge and then you separate by size. So what I want you to do to see if you understand this concept is I want you on a piece of paper, if you have one available, or on anything really, draw a square. And on the bottom left corner, we're going to put pH 1. In the bottom right corner, we're going to put pH 14. I want you to try and determine for these 5 proteins, for these sizes and PIs, on this square, tell me where they're going to be found. Predict where they're going to be found on 2D gel electrophoresis if they have these characteristics. All right. So I'm going to give you a couple minutes to do that. See if you understand how to separate by 2D gel electrophoresis. If you have questions, please let me know. Otherwise, yeah, I'll give you a couple minutes to do that, and I'll come back with what I got for this answer. do about one more minute for this. All right, so let's see if we can figure this out. So protein 1, PI of 5.3. So it's going to go to around where a pH of 5.3 is and then stop moving. 32 kilodaltons, relatively light, so it's going to travel far down the gel. So protein 1, it's going to be like right here. Protein 2, PI of 8, so it's going to be further on the gel than 1. Very heavy, so it's not going to move that far down the gel. It will be like up here. Protein 3, 13.2, so that's going to be very far to the right. So it's going to be almost where pH 14 is. 85 kilodaltons, so it's going to move farther down than protein 2. Protein 3 is going to be roughly right here. Protein 4 has the same PI as protein 1, so it's going to be in the same column. It is heavier than protein 1, lighter than protein 3, so it's going to be between those two. So it'll be like right here. Then protein 5, it has the same weight of protein 1, so it's going to be in the same row. Same PI as protein 2, so it's going to be roughly right here. And that is basically how 2D gel electrophoresis works. You separate by not only charge, but also size. And again, these are really good for what's called proteomics, where you just want to study a lot of proteins at the same time. Any questions about our 2D gel electrophoresis? All right, if not, that ends the material for test 1. And let's get on to the PowerPoint I had for today. So this test 2 also, since we have the test on Monday, I'm gonna do y'all a little bit of a favor. So the reading guide that's due on Monday, and I just decided this in the last few minutes. I'm gonna move that to be due Wednesday morning. So you have 2 reading guides due on Wednesday morning. So yeah, just as a way so you don't have to like focus on watching the videos for Monday if you rather spend that time studying for the test. So I will make that change in Blackboard this afternoon, where the reading guide's no longer due on Monday morning, but Wednesday. If you already did that reading guide, you don't have to worry about it. It's already turned in. All right, so what we're going to talk about in this section is that we're going to look at how to sequence a protein. That is, what is the amino acid sequence of a protein? And we're going to start with what's called the damsyl chloride method. And what danzyl chloride is, it's N group analysis. That is, we use it to figure out what the N-terminus, the first residue of a protein is. What is the N-terminus residue of a protein? And it's a rather simple reaction where we take our protein and we add the reagent called danzyl chloride, 5-dimethylamino-1 naphthenol sulfonyl chloride. That's why it's called danzyl chloride. It's way easier to say. And danzyl chloride in the will react with the N-terminus of amino acids to get this danzyl polypeptide. You then add acid and very high temperatures. So it's a very dangerous experiment, but you can do that. And the amino acid will pop off. In fact, you break every single peptide bond when you add acid and high temperature. You will then be left with one amino acid that is fluorescent. And this amino acid has a different wavelength based on what it is. So all danzyl amino acids have different wavelengths. So lambda's wavelength based on what amino acid has been danzylated, right? So you can determine, okay, what is my first amino acid? It's an alanine, cysteine, glycine, et cetera, et cetera. So this method is just used to determine what is the very first amino acid. However, I would say there is another purpose for doing this, and it's not just for determining what the first residue is. Can anyone hypothesize why might you do this danzyl chloride method, not just to sequence the N-terminal residue? Like, what is it? What could be another use for this? And it's not obvious. This is one of those tricky ones that I'm not expecting anyone to be able to answer necessarily, but I'm just wondering, can anyone think of what might be another purpose for doing this method? Ideas, please. Here's a hint. Proteins can be made out of multiple peptides. Alter N groups to stick them to each other. That's a good idea. Unfortunately, that this method won't do that because at the very end here, right, we chop all the amino acids up. So I like that idea, but unfortunately, danzyl chloride can't stick peptides together. I don't want to have any other ideas based on that hint. While I have dogs barking all over the place. Does it give them the opportunity to rearrange? Unfortunately, after you make this cut, there's no going back. So your protein's cut forever after doing this. Good idea, but yeah, you can't unscramble the egg once it's been scrambled. Modify other amino acids. So danzyl chloride could modify arginine. Unfortunately, at the end of it, you're going to chop it up. So good idea. Unfortunately, won't be used for that. Could degrade harmful proteins. It could, yes, for sure. It would do that. But it would only do that in like a test tube. Like it would have to be outside the cell to do that. So, I mean, we could definitely do that. But it wouldn't be exactly beneficial for us to do that. But again, all good ideas. I like that people are thinking, and that's really what I want from these type of things. And honestly, that's the main purpose for people going to college, I feel, is to train yourself to think and think about scenarios like this and come up with new ideas, right? That's what a college education, my experience, best prepares you for. It's teaching you how to think and learn. So what this can be used for, and it goes along with my hint. Let's say I have a new protein. I discovered a brand new protein. I don't know what I'm drawing those random lines for, but this method is to determine how many peptide chains are in your protein. And this is something we'll talk about when it comes to quaternary structure. But every peptide chain has an N and C terminus. Multiple of these chains can be combined to make one protein. So the danzyl chloride method could tell you, okay, if I have a brand new protein, how many chains is it made out of? So that's just another way to think about this. No worries. Thanks for letting me know. Right. So usually the danzyl chloride will sequence the N-terminus, but it can also tell you how many peptides are in a protein as well, because each peptide has an N-terminus. So let's talk about how we sequence a protein. And we are looking here at a protein made out of two different peptide chains. We have a blue chain and we have a purple chain. And these chains are connected by a disulfide bond. So the first step is to break any disulfide bonds in your protein. We already talked about how to do this, 2-mercaptoethanol. So the first thing you want to do if you have a protein, you want to figure out what the amino acid sequence is that you add 2-mercaptoethanol. After that, you break your proteins apart. And you do this multiple times. You do different breaks. So that's what's being shown here in 2A and 2B. We use one reagent to get breaks like this. And in 2B, we use a different reagent to get breaks like this. So you have multiple copies of your protein in different test tubes. And you just use different reagents in these different test tubes to get different breakages. So that's the first two steps if I want to figure out the sequence of an unknown protein. 1, break disulfide bonds. 2, chop up my proteins in different ways. And once you chop up your proteins in different ways, you need to figure out the sequence of each section. So I need to figure out, okay, each little mini section I have, what amino acids make up that sequence? And the way you do this is called Edmond degradation. And this is an automated process which loops over and over and over again. So let's look at how we figure out the amino acid sequence of a protein using Edmond. Here, our reagent is called PITC, phenylisthocyanate, or PITC. And like the previous technique, the danzyl chloride technique, the first step's the same. Our reagent here, PITC, binds to the N-terminus of our protein. But the next steps are different. After that, we use anhydrous trifluoroacetic acid. So we don't add water, so no water. We add pure acid. And what happens is that the amino acid that's attached to your reagent will cyclize and form a PITC amino acid. Or sorry, not PITC, PTH amino acid. This is fluorescent. And so you measure the fluorescence of this PTH amino acid. Each amino acid has a different signal, so you know what amino acid is on your N-terminus. However, the power of this process is that all the other peptides don't break. All the other peptide bonds remain intact. And so only the first amino acid comes off. And so you just repeat this process now. So you remove the PTH amino acid. You have the rest of the amino acid intact minus one amino acid. You add your reagent again. Repeat, get a new amino acid. Repeat, repeat, repeat. And this way, you are sequencing one amino acid at a time from your protein. And you just figure out, okay, what is the amino acid sequence of my protein? So that's called the Edmond degradation method for peptide sequencing. Is there any questions about that process diagram right off the bat? Any confusions or anything you want me to go in more detail on or anything at all? All right. So let's make sure we have that. Let's make sure we understand that. So I have a peptide, ser, cis, lu, vel, asp, and I do Edmond degradation to it. After cycle 1, what am I going to make? So this is multiple choice, A, B, C, D, E. Take a minute and see if you can get the correct answer. If you have questions, let me know. I would love to say I had a poll for this, but apparently I skipped over this question in making my polls this morning. So I don't. So just answer this one to yourself and see if you get the right answer after I come back in a minute. All right, so with Edmond degradation, you only remove the N-terminus while all other amino acids remain the same. That would be E. You're going to make your PTH serine, and the rest of the amino acids will stay together in their own peptide chain. And so that's round one. And then round two, you would make like PTH cis and be left with Lu, Val, Asp. Round three, PTH Lu, then BD. Then you do that to make PTH Val, and then PTHD at the end. Whoops. So that's Edmond degradation and how we sequence proteins. And with that, that brings us to the idea of protein evolution. After sequencing the same protein from different organisms, we can start to take that information and determine how proteins have evolved over time. And what we're looking at here on the left is we're looking at the sequence of homologous proteins. And what homologous proteins are is that they're proteins in different organisms that basically have the same function. And so here we're looking at the protein called cytochrome C. And we've just listed or lined up the primary sequence from many different organisms, from mammals, vertebrates, insects, fungi, and plants for cytochrome C. And when you do this and you look at the primary sequence, you get certain, you can find out certain things. One, you can see which amino acids are called invariant residues. For example, at this position, you have nothing but glycine. No matter what organism you look at, it's always going to be glycine. At this position, position 14, you always have cysteine. Doesn't matter what organism you look at position 14 for cytochrome C, it has to be cysteine. Position 30 has to be proline. What this tells you is that that amino acid is critical to the function of that protein at that position. That means at, like, for example, position 14, if you have a mutation that changes that cysteine into something else, then that cytochrome C no longer works. Otherwise, we would have seen that cysteine be mutated away in some other organism. So that's the idea of an invariant residue. When you combine the sequences of different organisms, if that residue never changes, that residue must be critical. That exact amino acid must be critical to the function of that protein. Another type of relationship we can see is called a conservative substitution, where it doesn't matter we have a specific protein there. What matters is that we have the same type of protein. And so at position 21, you can see that we either have glutamic acid, E, or aspartic acid, D. So what this tells us is that at this position, all that matters is that we have a negatively charged amino acid. It doesn't matter if it's D or E. All that matters is that it's negative. Here at position 42, it's the same idea. We have Q, S, or T. So it doesn't matter what amino acid's there. All that matters is that it's polar. So that's a conservative substitution, is that if we look at all the different organisms, as long as the same class of amino acid is there, that's all that matters. Then you have hypervariable mutations. That's where many residues can be substituted. And what that tells us is that it really doesn't matter what type of amino acid you have there, just that you have an amino acid. For example, at position 11, we have some hydrophobic amino acids. We have a polar amino acid, and we have charged amino acids. So at position 11, it's not that strict what type of amino acid you have. And this would be an example of neutral drift, random mutations that don't significantly alter function. So these are just mutations that's happened over the millennia for this protein, but it seems that the protein still works. Another example would be at position 3 here, where you have both polar and non-polar amino acids appearing again. And position 4, you have negative and positive amino acids and polar. So it really doesn't matter what's there, just that you have something there. And that is called hypervariable mutations, or residues rather. And so just to make sure we have those ideas down, and we know our amino acid classes, I have a question here, and I do have a poll for this one. I want to know first, which of those amino acids, and there's more than one answer, which amino acid would be a conservative mutation for Val? So I didn't make it multiple choice, so just pick one, but there are multiple answers that could be a conservative mutation for Val. For 2, which amino acid that I've shown there would be a conservative mutation for arginine? And C, we're going to skip due to time. Conservative mutation means it's a mutation that has the same characteristics. So it's a different amino acid, but it's the same type of amino acid. That is, it's still a hydrophobic amino acid, or it's still a polar amino acid, or still a positively charged amino acid. So when you look at the primary sequences of different organisms, if at that position you have the same class of amino acid, that's an example of a conservative mutation. All right, so let's take a look at this. So valine is a nonpolar or hydrophobic amino acid. So to be a conservative, so nonpolar or hydrophobic. Two words that mean the same thing. So a conservative mutation of val would also be amino acids that have the same characteristics. So alanine is nonpolar, so that could be a conservative mutation. Leucine is nonpolar, so that would be a conservative mutation. Glutamic acid is negatively charged, so that won't be. Isoleucine is hydrophobic, so that's a conservative mutation. Asparagine is polar, so it wouldn't be. So arginine now is positively charged. So for a mutation to be considered conservative, it would have to mutate into a positive charge amino acid. The only other positive charge amino acid is lysine. So the only conservative mutation of arginine would be lysine. Any questions about that idea of conservative mutations? All right. The last exercise I have for you today is to take that idea, but we're going to look at a fake sequence here where let's imagine I took a protein from the same protein from six different organisms and lined up their primary sequence. For every position, I want you to tell me, one, is the mutation, is it unable to tolerate substitutions? Or is that position only able to tolerate conservative substitutions? Or is that position highly variable? So just to give you an idea, position one, we have asp, asp, glue, asp, asp, asp. All of these amino acids are negative. So position one would be, that position can tolerate conservative substitutions or mutations. So last poll for today, last poll for the week is for each position, tell me if it's unable to tolerate mutations, if it can tolerate conservative mutations, or if it can tolerate highly variable mutations or substitutions. So try to get that down. If you have questions, let me know. All right. Time's running out here. Looks like, all right. So for some positions, we are having, not quite sure. Other ones, we're good. So let's take a look at all these. And this is really, it becomes a lot easier once you have like your amino acids memorized, so you know which amino acid is what type. So isoleucine, nonpolar; leucine, nonpolar; alanine, nonpolar; phenylalanine, nonpolar. So this is conservative as well. No matter what, at position 2, all that matters is that you have a nonpolar amino acid. Doesn't matter what it is. While position 3, these are nonpolar. This is polar, and this is negatively charged. So this is highly variable. It doesn't matter what amino acid is there. You just need an amino acid. Same thing with position 4. Negative, polar, nonpolar. So that's highly variable. 5 is interesting. I would say histine's more polar than charged. So I would say this is conservative. You can make the argument that histine is positively charged, so this might be more highly variable. But I would be more comfortable saying this is just conservative, since histine is more likely to be polar than charged. 6, I think everyone got that one because 6 is the easiest. Invariable, you can't have any mutations. Same for 9, that's invariable. 7, polar, negatively charged, nonpolar. So that's highly variable. 8, negatively charged, negatively charged. But then you have this nonpolar thing hanging out. So this is an interesting case in that this can happen. This happens quite a bit in the real world. Going by our actual discussion, this would be highly variable. But you would really want to study the tuna protein to see why there's an L there. Because it could be, this is actually unable to tolerate substitutions, but tuna is just an oddball. So if you looked at like other proteins, maybe everything else is glutamic acid, and tuna for some reason isn't. But just to go on our classification as we're learning them at the beginning here, we're going to say this is highly variable because you have negatively charged and a nonpolar residue. Leucine, isoleucine, nonpolar. So these are all conservative mutations. And again, these are all this type of information is found by looking at the primary sequences of homologous proteins in different organisms. So any questions whatsoever here at the end of our discussion for today? All right, if not, remember our test 1, we're going to have that starting at 9. So I'll open it up at 9, and then I'll probably open it up like till 11. You'll have 1 hour to do it, and it'll be found under Blackboard. I'll probably make a little video once I have the test made to show you how to access that, but I'll also be available as well. So for this class, H is always polar. Histidine's weird because the pKa of histidine is 6. So it's both polar and charged, positively charged. So we'll see moving forward that we can't just say it's always polar. It depends on the protein. So we have to do that by case situation. For this type of thing, where we're just like not looking at the protein, just looking at the sequence itself, it's a lot easier to call histidine polar. The charge is based, it's really, you have to look at the actual structure of the protein to determine what histidine is. So yeah, it's one of those cases where there's no easy answer. Well, we have class after the exam on Monday. You'll have class before the exam on Monday. So from 8 to 9, we'll have like regular class, and then I'll let everybody, then the exam will open up, and yeah, we'll have you take the exam. I'll still be online if you want to ask me questions about one taking the exam. It's like, like in a normal classroom setting. So I'll be on Zoom if you want to pop in, and I can put you in like a private chat or something like that. Yeah. Like I said, I haven't made the test yet, but once I make the test and finalize like the exact format I want, I'll probably make a little video and put that online to let us know what's happening, so you have some time to mentally prepare yourself. But yeah, if you want to have a meeting, go over some things, shoot me an email, stop on by, and we can have any discussion you would like. Otherwise, study hard, have a good weekend, and I will see everybody on Monday, if not sooner. Take care, everybody.